Hello dear students, welcome to Practically. We bring learning alive, and in today's session we are going to see five most important questions from the chapter Biotechnology Principles and Processes. Before to this, we have already seen five important questions from the previous two chapters for term 2 biology CBSE exam, and in this chapter we are going to see the five important questions from this chapter, which is actually very important in itself. So stay tuned and watch till end. So let's begin with the first question. With the help of diagrams, show the different steps in formation of recombinant DNA by action of restriction endonuclease enzyme EcoR1. So, as this chapter is biotechnology process and principle, this question belongs from the topic of tools in recombinant DNA technology. So, let's see how to answer this question step by step. So yeah, first we need to tell that what is a restriction enzyme. It is basically an enzyme which is going to recognize specific palindromic sequence, nucleotide sequence in DNA. And we know that the recognition sequence for EcoR1 is GAATTC, and the same from the opposite side. This is from 5' to 3' end, and from the opposite side we'll have CTTTAG. So just remember this much sequence, and this only have to write ulta from this side. Okay, simple. Now, restriction enzyme is going to cut the DNA strand at certain points from the palindromic place. For example, in case of EcoR1, it cuts the DNA from between G and A. As you can see here, this is the point where it is going to cut the sequence. Same will be from the other side, on the other template, other strand, second strand of DNA between G and A. It will cut over here. So the ends produced will be sticky ends. Okay. So this G got cut, this AATTC got on the other strand, and similarly it will be there here and here. So these are called as sticky end, which is going to stick like this. So EcoR1 is a enzyme which produces sticky ends, and then finally they are joined together with the help of enzyme ligase. So yeah, so that's what we need to tell, that EcoR1 cuts the DNA between the sequences G and A only when the sequence GAATTC is present in the DNA. Okay, so that is why they are called as specific site. Okay, they recognize specific site, and only at specific location it is going to cut. Yeah, and it is going to produce sticky ends. So, and they will be joined later by the enzyme DNA ligase. So this is all you will mention in this answer. Okay, simple question. Now, let's see the second most important question from this chapter. Explain methods of vectorless gene transfer. Again, a question from tools of recombinant DNA technology. Let's see how to answer. Yeah, so the first method mentioned over here is chemical method. Okay, so in chemical method, what we are going to do? First, you can also write why we need this method, because we know that a cell cannot uptake DNA. Okay, the surface of the cell, it is made out of what? It is made up of lipid bilayer. Okay, so this lipid bilayer, lipids we know they are how? They are hydrophobic, right? But DNA is a hydrophilic molecule. So because of the difference in the nature, the DNA molecule cannot simply pass through this. Okay, we'll have to make the cell competent to take up the DNA. So that is done by various methods. The first method out of that is the chemical method. Okay, so what we do in in case of so DNA molecules can be forced into enter host genome only in those cells which do not possess cell wall. Okay, in that only you can directly put, but if it has cell wall around it, obviously it's not that easy to transfer DNA. So in such case, what we are going to do is the naked plant protoplasts are mixed with molecules of linearized plasmid DNA containing the foreign gene. Okay, and then these two are mixed in medium having rich magnesium ions, Mg2+ ions, in place of calcium ions, following which 20% polyethylene glycol, PEG. This is a chemical which is added, and then the treatment is given, which is going to promote the frequency of transformation. Okay, so this is how you will enter do the chemical method to for the vectorless gene transfer. Okay, so in the same thing, the second method of vectorless gene transfer is electroporation. Okay, in this we are going to use the plant cells having the cell walls, protoplast, in a solution containing linearized recombinant DNA plasmid, and that mixture is then exposed to low voltage long pulses or high voltage short pulses. So that is what the electroporation will be done. This helps to like create transient pores, induce transient pores, so that the DNA molecules can pass through that. Okay, so this is again the process of electroporation. The electric shock is given by high voltage or short voltages, and then that will open up the pores to take up the DNA. Next, particle bombardment gun method. Okay, so yeah, so this is an additional information. You need not write so much of details in it. That this method was developed by Professor Stanford and co-workers at Cornell University. This is like just additional information. What main thing you need to write is this method is also known as ballistic technique, in which what we do is the gold or tungsten particles are coated with DNA molecules and then are shooted into the plant cell, okay, using pressure. So basically, as you can see, this is the gun by which having tungsten or gold particles, which are, so because those are very hard, they can easily pass through the cell wall, and having the DNA particles, they are directly injected, like put into the cell. So that is the method of gene gun. And the last method is micro injection, in which directly we are going to inject the DNA into the cell. We are going to hold the cell on place and then using injection. Obviously these are very tiny, on a small scale they are done. They are going to be injected into it using micropipettes and micro syringe. Okay, so this process is mainly used in animal cell. Okay, but it also can be applied for plant cells as well. Okay, so these are the 4 various methods for vectorless gene transfer. So just we need to explain it in short depending upon the marks the question carries. Okay, moving ahead to the next question, describe the roles of heat primers and the bacterium Thermus aquaticus in the process of PCR, polymerase chain reaction based question. This is, let's see how to solve them. If any one of you can think of the answer, please do comment in the chat box, or you can also leave a comment by which we can know. Try to answer, try to attempt. This is a question based on PCR, that is polymerase chain reaction. Okay, let's see how to answer. Yeah, so they have asked roles of 3 things: heat, primers, and the bacterium Thermus aquaticus. Okay, yeah, so role of heat, heat helps in the denaturation process. So we know that there are 3 processes involved in the entire PCR. First one is denaturing, second is annealing, and third is extension. So the heat plays a very important role in denaturing process, where the double stranded DNA is separated. Okay, and that temperature is around 95 degrees Celsius, so that the both strands separate. Then primers, what do primers do? They are basically oligonucleotides, small oligonucleotides, which bind to the complementary region of template DNA and helps in extension of new chain. Okay, and role of bacterium Thermus aquaticus. Now, why we use this particular bacteria? Because it produces thermostable Taq DNA polymerase. So we know that for the DNA replication, we need DNA polymerase enzyme, but we know that enzymes are very sensitive to temperature. Now, the normal DNA polymerase, it won't act at this high temperature, because in this entire PCR process, we are using a very high temperature as per the requirement of the process. So, we need an enzyme which can sustain at such high temperature. So, Thermus aquaticus is a type of bacteria, as the name says, Thermus aquaticus. They are found in the hot pool places which are present. So they naturally are present in hot atmosphere, so they have enzymes which can bear that temperature. So that's why we use these particular polymerase which can tolerate high temperature and forms new strand. Okay, so this is what the answer for this question. Here is the next question. How has the use of agrobacterium as vectors helped in controlling Meloidigine incognitia infestation in tobacco plants? Explain in the correct sequence. Okay, let's see how to answer this. Yeah, so Meloidigine incognitia is infects the roots of tobacco plants mainly and causes the reduction in yield, and for this the RNA interference method was introduced. So we have to explain that in short, where we introduce nematode specific genes, okay, into the host using the agrobacterium vector. So agrobacterium, naturally it infects the plant roots, so we are using the property of that bacteria to kind of deliver the nematode specific genes into it, and that will produce both sense and antisense RNA in the host. Okay, so now sense and antisense RNA are complementary. Being complementary, they are going to finally make double stranded RNA, and double stranded RNA cannot undergo translation to produce the functional protein. So the process of translation will be stopped, protein won't be produced, and basically it will get silenced. The mRNA will get silenced, means it won't undergo further process. So if the proteins are not produced, obviously the nematode won't be able to survive. Okay, so this is how they get protected from the parasites. Okay, got it? If any doubt in any of these questions, please do leave a comment and we'll be there to help you out for further preparation. Next, let's see the next question. How has the development of bioreactor helped in biotechnology? Name the most commonly used bioreactor and describe its working. Okay, fine. So this is a question based on the last part of the chapter, that is the bioreactors. So yes, anyone, if they know the answer, you can just give hints what all things should include in it, or else we are going to anyways discuss the answer. Let's see. Yeah, let's answer the first question. How has the development of bioreactor helped in biotechnology? Okay, hello PUBG player. Yeah, because you might not be able to see me because we have to cover the answer, we have to show the answer. So that's why I have to move away, because it's important for you to see the screen. Okay, so screen is important, the answer is important. So let's focus on that. Let's focus on the keywords. You're all anyways hearing my voice. Okay, so if you have any doubt or query, you can ask me. I'll come in front of you and answer. Okay, I hope PUBG player your query is solved. Let's discuss this important question over here. How has development of bioreactor helped in biotechnology? Okay, and if you have anything to say or ask, please do type in the chat box, PUBG player. Yeah, so see, bioreactor provide large volume, okay, because small volumes will not produce enough quantity of products. We need large volume, so for that we need a special big vessel for the reactants and the material to be processed, and that's why for the products in large quantities bioreactors were introduced. Okay, So that is how it has helped the biotechnology. So obviously they contain biologically convert the raw materials are biologically converted into specific products based on animal or human cells or the particular enzymes. And what is the role? This is very important. This point is important, that bioreactor provides optimal condition for achieving the desired product by providing optimum growth conditions like temperature, pH, substrate, salts, vitamins, oxygen. Okay, these all things are required for the bacterial or any whatever microorganisms you are trying to grow, which is going to give out the necessary products. For that you are going to need, so bioreactors provide these things in it. Okay, I hope you have understood. So for this answer, these all important keywords and points should be covered. Okay, so I hope whoever are watching, they are with us. Is it PUBG player? If you have any doubt, please do comment or give me a thumbs up so that I'll understand you are understanding, those whoever are watching. Yeah. Now moving ahead to the second part of the answer. Second question was, name the most commonly used bioreactor and describe its working. Okay, let's see. So most commonly used bioreactor is of stirred type. Okay, stirred type bioreactor. Actually there are two types of bioreactor. One is stirred type, one is the sparged, air sparged bioreactor. So stirred type bioreactor, it is cylindrical, having a curved base. As you can see, just draw this diagram. For this question, drawing this diagram is important. So do practice this diagram, okay, of drawing this bioreactor. To just draw the cylindrical shape, you have to draw the impellers in it and the motor and the pressure gauge and openings for the steam and acid base control. Okay, and a inlet over here for sterile air. Okay, so a stirrer facilitates mixing and oxygen availability, and then it also consists of various parts like agitator system, which helps in mixing the things, oxygen delivery system, as you can see here, the sterile air is delivered into it. It also has pH control, you know, so that the pH should be maintained. It also has foam breaker. Why do we need foam breaker? Because when they rotate, in certain cases it can cause foaming. So if it becomes too much of foam, it can affect the function. So this will break the, otherwise the foam can come out also through this thing. So it will break the foam so that it becomes even and it doesn't come out. Okay, so these are the types of bioreactors. Okay, now, so these were the 5 most important questions from the chapter Biotechnology Principles and Processes, and these are some important topics which you should specially focus on while studying this chapter, because a lot of questions are expected from these topics. They are important. What are those? Restriction enzymes from the tools section, also cloning vectors. Many students find this cloning vectors little tricky. So don't worry, we have made a one-shot video in which we have explained this, and there is one more video on tools of recombinant DNA technology. So do check it out on Practical 11th and 12th channel. You will surely find helpful videos and content for your preparation. Then PCR and bioreactors. So these are the important topics which you should focus if you have to score well from any question asked from this chapter. Okay, so if any doubt, any query, do leave a comment below this video and we'll be there to help you out. Thank you for watching today's session, and we are also going to come up with 5 important questions from remaining chapters as well. So stay tuned with us, subscribe to Practically, and see you all in the next session. Thank you.